So we're going to start chapter 36 by looking at chapter 36.1. I listed all the standards that we need to go over for this unit. But 36.1 is looking at studying populations in terms of their size and where they are. So remember, a population is a group of organisms of the same species. And we talked about like the evolution of populations, and we defined what a population is again in chapter 35. Demography is the statistical study of populations, including humans. You've probably heard of demographics before. It's another very closely related word. Population size and population density. Population size is how many individuals are within a population. Population density is the number of individuals within a specific amount of space. So how many individuals per area? Population size. Regarding population size, larger populations tend to have more genetic diversity. We talked about, for example, the bottleneck effect or the founder's effect, where the resulting population has less genetic diversity than the original population. And therefore, even if that population rebounds, it will have less genetic diversity than it did before. That's really important because as there's selective pressures in the environment that change, the more genetically diverse the population is, the more likely it is to sustain those changes and be able to survive and reproduce based on certain members in the population being more likely to have an adaptation for that change. And regarding population density, smaller organisms tend to more densely distribute themselves. Okay, so cockroaches tend to be very densely distributed. Yuck, right? And so whales, on the other hand, tend to be calling each other from miles away because they're really big, so they're not as densely distributed. And so this shows there is a mathematical relationship between the size and the density of populations, and you can see all these different examples. The bigger the animals, the less densely they tend to be populated. That makes sense because more biomass requires more energy to sustain. When determining population size, why not just count every individual? Why don't we, if we want to know how many blue whales are in the world, why don't we go out and count every single blue whale? Well, that's not very practical, is it? Right? Hopefully you can realize it's not very practical. Or why don't, if you want to know how many cockroaches are in Dakar, shouldn't you just try to find every single cockroach, go ahead in every person's apartment? No, right? That's not going to work. Or whatever, how many little brown birds are in Dakar? I don't know the name of those birds. I should know, right? But you can't count every single one. So instead, one way to do this is to use a quadrat, where you basically have areas of the population taken and use this as a sample and say, okay, how many individuals do I see in this area, in this little area, and say, well, if I have reason to believe that the density in this little area is the same as this larger area, then I can determine what the density is and use the density and the size of the big area to determine roughly what the population is. Okay? So for example, in a large field, a scientist could measure the population of some plant or bug or something and say, okay, I see this many right here, so now I can estimate how many total are probably in this much larger field or this much larger area. And this could be used not just with little bugs or something, but with deer. Maybe look at a larger area and find how many deer are in that particular area at any single time, but then you use that to estimate the deer population in a much larger area than you could feasibly scan all of. Okay, so here's an example. Scientists wanted to know how many deer are in the town of Hamilton, so they looked at these specific areas within Hamilton to get an idea of overall what the density is and therefore how many deer overall there might be. Another method besides quadrats is mark and recapture. So this is capturing some of the animals and then releasing them and then recapturing animals and determining how big the population is based on the percent of animals captured in the second sample that were captured the first time. So you mark the animals with a marker or something, and then you release them. Then there's a second round of sampling, and the higher the percentage of recaptured animals, the lower the size of the population. Because that means, okay, there's not that many total because you're capturing the same ones you already got. Whereas if you had a very small recapture rate, that would mean you're not capturing the same individuals because the population is very, very large. Here's another just picture of animal that was captured and then released, and then they could see if they collected that same animal again or a different one. And so this is the mathematical relationship, and I don't think mathematically you need to be able to like plug and chug to determine this, but you need to conceptually understand the idea here that we're looking at how many were captured the first time, and then we can compare that to how many were captured the second time that were captured the first time. So I wrote it like this. So let's say there was 100 in the second sample, 100 in the second sample of some population, and you divide by 20, which were recaptured, and then you multiply by the size of the first sample that was captured, or how many were captured the first time. Again, the mathematical details here aren't super important. I wouldn't take super long looking at this. As far as I can tell, I don't remember AP Bio ever assessing on this directly. But the idea is that the higher frequency of recapturing, that means the population's smaller. Because there's less members, you're more likely to recapture a member you already got in the second sample. Of course, this could be limited if the captured animals are more or less likely to be recaptured. If for some reason the tags made them more or less likely to be recaptured, or if the animals that were captured the first time learned and realized, I'm not going to go in that thing again, right? That will impact your results, because mark and recapture works the best when the originally captured animals have the same chance to be captured as the animals that weren't captured the first time, because that's the whole idea. So let's test your understanding. Hypothetically, let's say a tag on a mouse makes it more likely to be noticed by predators. How will this affect population estimates by mark and recapture? Go ahead and take a minute to think about it. Hopefully you took a minute to think about it. And you should say, A, less mice will be recaptured, so it will seem to be a smaller proportion of the population than it actually is because the recapture rate is too high. Species dispersion patterns shows the spatial relationship between members of a population within a habitat at a particular point in time. And so we could have uniform distribution, like these penguins here. We could have random distribution, like these weeds, or clumped distribution, where all the organisms in a population stay together. Life tables looks at the age of organisms in a population and how many of the individuals make it to each of a certain age, and often differentiates by gender as well. And mortality rate is the percentage of surviving individuals die in a particular age interval and their life expectancy at each interval. Okay, so what's the chance that this particular organism dies in this particular year? So for example, we could be looking at this particular sheep species and seeing, okay, so what is the number out of 1,000 dying in 0 to 0.5 years? And then what's the number 0.5 to 1? And you can see that 0 to 0.5 and 0.5 to 1, the mortality rate's higher, meaning a higher percentage or a higher chance that they die at that young age. And then the mortality is way down from 1 to like 7 years, and then it starts to go way up again until there are no individuals left and the mortality rate is really high. Okay. And so like you can see that 11 to 12 years old, for example, the mortality rate is 937. That doesn't mean that 937 out of 1,000 in the population die in that year of life, it means that if you had 1,000 at that age, 937 would die. In other words, any individual has a 937 out of 1,000 chance of dying in that year, which is like a 93 or 94% chance of dying. Okay, that's what that is saying. And different organisms exhibit different chances of survival or different mortality rates at different times. And so that's shown here. Type 1 is where there's a low mortality rate until suddenly there's a really high mortality rate and just all the organisms die. Type 3 is the opposite, where not many organisms make it very long, but the ones that do will keep going and going and going, and there's not a drastic drop-off or not a dramatic decrease in mortality rate at some point. And then type 2 is somewhere in between. Okay, so here's another example of this. You can see type 1, the mortality rate is pretty low, and then it increases drastically at a certain time. Type 3, the mortality rate is really high at the beginning, and then it decreases through time, and type 2 is in between. Okay, so like the clownfish, if you've seen Finding Nemo, that's a type 3, where all the individuals die really quickly, and then there's a few who survive for much longer. That is it for 36.1.